Laboratory Scalable Tissue Imaging and Modeling with David Mayerich.
My name is David Mayerich, and I'm an associate professor in electrical engineering at the University of Houston. I'm going to give you a brief presentation and tour of the Scalable Tissue Imaging and Modeling Lab here at UH. Our research focuses on the acquisition and data mining of tissue images at scale, enabling the generation of whole organ, cellular level tissue maps. This includes the development of instrumentation, such as high throughput microscopes that can collect three-dimensional images of cubic centimeters of tissue at submicron resolution. This is cellular level resolution. My personal background is in computer science, and we do a significant amount of high performance computing focused on analyzing massive amounts of image data using heterogeneous hardware like graphics processors. Researchers that come through our lab leave with expertise in high performance computing and the ability to program in C++, Python, and CUDA. Finally, we develop methods for visualizing tissue maps by leveraging research in computer graphics and volume visualization and sparse encoding to deal with these massive volumetric data sets. We currently have research projects focused on adapting some of this work to virtual reality using publicly available software platforms like the Unreal Engine. Here are some examples of the types of images that we can acquire. These were collected using an earlier technology, knife-edge scanning microscopy. So this image is a cross-section of a mouse brain where the vascular system is perfused with an opaque ink. What we're doing is collecting cross-sections of this vascular network and assembling them into a three-dimensional model. This allows us to build comprehensive models of microvascular networks that exceed the size and resolution of what you can acquire using current technologies such as light sheet microscopy. Now, the technology that we're relying on is based on some recent work out of UC Davis, where they've shown that deep ultraviolet excitation induces fluorescence in common labels, but also limits the penetration depth into the sample. So what this allows them to do is acquire high-quality images of tissue histology without having to go through the time-consuming process of tissue preparation. So you can do this with fresh tissue. Now, we've leveraged this technology for three-dimensional imaging by adding a microtome component, essentially a knife, that ablates image tissue, revealing the block face underneath what we've already imaged. So by repeatedly imaging and cutting, we can create high-resolution images without any constraints on tissue depth. So here's an example of a vascular network acquired using this technique. So once we've collected the images, we can stack them on top of each other to build a digital representation of the vascular network. Now, moving a step further, we're also using high-performance computing to train neural networks to identify and segment components of the sample. So in this case, we have a 0.5 millimeter cube of tissue along with the segmented cellular and microvascular components. And this was done using a neural network that we developed specifically to be efficient enough to work on three-dimensional data sets. Now, our ultimate goal is to design methods to mine these feature-rich data sets, allowing researchers to quantify and compare whole organ samples at the cellular level. So now I'm going to give you a walkthrough of the STIM lab.
So today I'm going to give a brief tour of the computing and imaging portions of our lab. We also have a new histology and multiplex imaging facility that is under construction, so hopefully I will be able to give a tour of that next year. So here we have an office space that is it is equipped with workstations for up to around 13 students. Each of the workstations are designed to facilitate parallel computing, so they're all equipped with CUDA-compatible GPUs. We also have a VR setup for some recent some visualization projects that we're working on for three-dimensional tissue visualization. And over here, behind this office space, we have a lab space that's designed for optical imaging. We have a 3D printer, an SLA printer for printing out optical components and sample holders to facilitate this research. So as far as imaging systems, we have several microscopes. The first one I wanted to talk about is a 3D confocal imaging system. So this is a resonant galvo system. So it's designed to be able to image very quickly. We have optical objectives that are very large working distance for imaging very thick samples, and like those that you would get with clarity or using techniques like expansion microscopy. This imaging system is an Agilent FTIR microscope. So this is an infrared imaging system that we primarily use to image two-dimensional samples, usually tissue biopsies. And what FTIR provides is a spectrum that allows us to characterize the molecular composition, and it also allows us to detect chemical constituents that are difficult to stain and image using a confocal or fluorescent microscope. So these are like small molecules and metabolites, and we've been primarily using this either for tissue histology, looking at methods to augment traditional tissue imaging using standard staining, but it's also a source of a lot of collaborations where we're looking at different materials to try and characterize conformational changes in molecules or different types of polymers. So over here there are a couple of other instruments. So this is a Nikon TI Eclipse. So this is a two-dimensional imaging system, traditional inverted microscope, but it also has a confocal unit that is a galvo confocal unit. So this is significantly slower than the resonant galvanometer scanner, but it is it allows us to image weaker signals. So signals that are less fluorescent, or maybe if we're looking at autofluorescence, it allows us to control the speed at which we're imaging a lot better. And this is a dye-SPIM system, so it's a dual beam light sheet microscope. And this type of system is designed to image larger samples, and it is basically a standard light sheet microscope with two beams that allow you to scan back and forth in order to, with some post-processing, you can reconstruct a sample at significantly higher resolution. So light sheet microscope has a disadvantage of resolution along the z-axis. This type of system allows you to compensate for that and get more isotropic resolution for three-dimensional samples. Finally, we have one of the imaging systems I mentioned in the presentation. This is a prototype MOVE system, so this is the UV tissue sectioning system. We've also got another prototype in the histology lab, but what this consists of is a three-dimensional stage and an optical system that is designed to excite with UV illumination, and then it collects emitted photons from fluorescent molecules on the surface of the sample. And then there is a microtome component where the stage can slice off the top section of the tissue sample in order to reveal deeper sections for imaging.
So I can, I couldn't be in the lab today, but I can take any questions.
So I have a very self-serving question. Sorry if I, so I'm just the reconstructions of the 3D tissue that you're doing, how possible would that be to do with like actual like human cortical tissue? And then my question became, is that saved as like a mesh, or how, because I'd love to then incorporate that data into some modeling of, the electrophysiology and that sort of thing.
Yeah, okay, so So the only issue with human tissue is that we have to introduce contrast somehow. So the technique that we're working on is very fast, and the speed and the resolution are the advantages. So we have to find some way to introduce contrast. We're doing that with a just injecting India ink transcardially. That's difficult to do with a human, but
that would yeah, they probably wouldn't let us do that.
no, but we are working on some labeling techniques. So we can stain an entire, say, a cubic centimeter of tissue using a technique called switch, which was developed at the lab. The postdoc that developed it was formerly at MIT, but he also developed Clarity. And so we've got a device that allows us to stain an entire sample. What we have to do is we have to find labels that survive the paraffin embedding process.
Okay.
It looks like quantum dots will. And so we're looking at quantum dots with lectins, which will bind to microvasculature. We're also working on other embedding protocols that allow us to do that immunohistochemistry. But once we get that, that's mostly an engineering problem. Once that's working, once we get appropriate labels, then there shouldn't be any problem with the imaging. As for the structure that's created, so technically it's a volumetric data set, a stack of images. In the case of that microvasculature, it's really high contrast, so it's easy to create a mesh from that. I say easy. You have to do a little bit more than thresholding, right? You have to do some intelligent segmentation. But
none of that is easy, I'm sure.
Yeah. But if I would also expect, if we were doing immunohistochemistry, for the contrast to be less, for there to be less contrast between, say, microvessels inside and outside, because just immunohistochemistry is a little more finicky. But I would not expect it to be a problem to segment, and resolution shouldn't be the issue.
Great, thank you.
All right, thank you, David.